protein synthesis. The mechanism of protein synthesis. Just as with DNA replication and RNA replication, here too we have three distinct phases that we separate. We have the initiation phase, followed by the elongation phase, and finished up with the termination phase. When we look a little deeper into the initiation phase, we see that we find a tRNA that can read the start codon, the AUG start codon. And we can see that right over in here. We see how we have that tRNA, and we then see how that reads that AUG start codon. And what it does, it brings a special methionine to the beginning the process, that then that methionine at the end will be removed. That initiator tRNA starts the amino acid chain with a special form of methionine, which will be removed at the end when the protein is done. Then, once we start that amino acid strand with that methionine, we can go to work and start reading that mRNA strand and put these amino acids once after another. So let's look a little bit at the elongation phase. During the elongation phase, we have a couple of different places at the ribosome that the tRNAs then attach and do their process. So we have also, if you can see down here, you have an A site, and the A site is where the incoming amino acid get brought in from the cytoplasm. So that tRNA attaches there, and then we got us the P site, and the P site is where we actively build the amino acid chain, and the polypeptides grow one after the other and build a peptide bond to attach each one another. And then we finish up with the E, sort of like an exit, the E site, and that's where the tRNA, the empty one now, it's not having any amino acid anymore. The amino acid is released, and so it's uncharged, and at that point it has to go back to the cytoplasm and get recharged, so to speak, to then be brought in at the appropriate time when we need to bring back in that specific amino acid. And so then the charged tRNA comes in and brings it in. All right, so let's write that down. So the A site is where the amino acid charged tRNA enter. Then the P site is where we bind amino acids to grow the polypeptide chain. And then the E site is where the tRNA is released to be recharged. And then when the protein is finished making, when we're finished with it, we're going to get us to the termination phase. And we finish with the polypeptide chain and can make the protein out of that when we reach the stop codon. So the stop codon releases the polypeptide from the ribosome. And then the protein is made, and the rest of how it then creates the different levels of structure. We've already worked on that in a previous session. Isn't it just fundamentally an amazing process of how we have us the gene go into the mRNA and then the mRNA go into the protein, how we're going to make the protein, those fundamental workhorses in our body. It's phenomenal. And then lastly now, we're going to touch on how genes are regulated. All right, let's talk about how genes are regulated. We're going to talk about gene expression. Gene expression. It is important for genes to be very well regulated when they should be expressed, when we need to make protein out of them. Cells need to make the right protein at the right time. It's the cells themselves that control which genes are going to be expressed. Because we don't express all the genes, just some of the genes are expressed, the ones we're needing. And gene expression is a complex process. And so it has to be very well regulated, because if it is mistakes go on and we make mistakes in the process, we can have disease come from that. Disease like cancer, for example. And what influences which gene is expressed? Well, we, for example, have very specialized cells like the muscle cells or the brain cells or the liver cells. And they're all specifically expressing genes to make protein that are necessary for their functions. Also, environmental factors and changes influence which genes are expressed. And lastly, fairly obviously, is when we have different stages in development, we also need to express different genes on those different times. So let's talk a little bit more in detail the places in the process where we can have control over the genetic expression. Firstly, in this situation, we have to separate the prokaryotes from the eukaryotes again. And the separation here is the nucleus. Because in eukaryotes, we have transcription happening in the nucleus, and then the mRNA goes out of the nucleus into the cytoplasm, and that's where we have translation. So we have distinct processes are separated distinctly. And so we have because of that, we have more places where we can control. Because on the other hand, in the prokaryote, where we don't have a nucleus, we have seen that the mRNA gets created, and as soon as it gets made, we're going to read the strand in the ribosome and make proteins. And so in that situation, and the mRNA is very short-lived. So also, as soon as transcription stops, not assume, but shortly after transcription stops, protein synthesis stops. So really, we have the protein synthesis is controlled mostly by what gene is activated and what gene is not activated. And so that is the prokaryotes have no nucleus, and mRNA is short-lived. And their protein synthesis stops when transcription ends. And since the eukaryote has that nucleus and transcription and translation is physically separated, we can have multiple levels of where we can have control of gene expression. So let's look at that and zoom in. So for example, we can have epigenetic. We can decide which genes is going to open, for example. And then we can have also a control at the transcriptional level. Are we making the gene or not? So from DNA to mRNA. And then we also have an RNA post-transcription level where we can control it when we put the caps on and when we take the introns out. And then when we go out of the nucleus and we make translation, we have another level where we can control. And then even more, we can also control the gene expression once it is translated and once the polypeptide is made at that point as well. So that gives us multiple levels of control. So let's write that down. Eukaryotes have a nucleus, so physically we can separate transcription from translation. And so that fact lets us control the gene expression at many stages. We have control at the epigenetic level, which is when the DNA uncoils. Then we can also have control at the transcriptional level, which is when the DNA gets copied into an mRNA. At a post-transcriptional level, when the mRNA is further processed, then at a translational level, once we get the mRNA strand read and make us a polypeptide. And finally, at a post-translational level, after the protein is made. Well, so that completes this entire process of molecular biology from the DNA all the way to the protein. So that's very interesting. And we'll see you soon again. Take care.